So thank you very much. Thank you very much to the committee to select our work and thinking that is deserving this award, and also for giving me the opportunity to come here and show you what the work that we have been doing in the laboratory of Eduardo Soriano in the last years, and this work relating a family of proteins that is related in regulating the mitochondrial trafficking in neurons. So first of all, let me introduce a little bit the work, the background. As you all know, mitochondria is a key organelle, has emerged as a key organelle in which a number of signaling cascades converge to keep cellular homeostasis. Mitochondria are the main providers of energy supply to the cell, and also in mitochondria converge a number of metabolic and biosynthetic pathways. It's also that's also an important role in calcium buffering, and in mitochondria has converted in main integrators and effectors or cell death pathways. For that reason, it's not surprising that mitochondria has been linked, or mitochondrial dysfunction has been linked to a number of pathological situations, ranging from metabolic syndromes, cancer, or more recently has been linked to almost all known neurodegenerative diseases. And why we need to find the reason of this in thinking why nervous system, or more specifically neurons, are such sensitive to mitochondrial function. Let me show you this image. This is an image of the first positron emission tomography done about 40 years ago, in which you can observe that the huge amount of glucose uptaken by the brain compared with other regions like heart or other regions of the body. This image, or we need to find the reason, is a point that indicates how expensive it is for the brain to keep neural system or neural activity working. The other main reason is the enormous complexity of the geometry of these cells that has processes that whose length or who has processes with who extend millimeters, centimeters, or even to 1 meter in the case of human motor axons. And with these two reasons, we need to find why neural activity is that expensive. And this is mainly trying in the main feature of neural system, that is the communication of these cells, the excitability of these cells. And this communication, I'm going to show just to take a representation in the dendritic spines, in these points where the two neurons are communicating, and the communication of these neurons means that when a neurotransmitter arrives, induce influx of calcium, high calcium inside the cell, and this cell calcium, that means the basic of neural communication, needs to be reverted, and for this the neuron needs to pump the ions back across the membrane to regain again excitability for the next income of neural activity. This process is a highly expensive process, and neurons need to have near these regions mitochondria, or a source of ATP or energy to supply the energy that is required to pump these ions back and to regain excitability. At the same time, it is known that mitochondria is able to uptake calcium, and thus is doing another function important in this process, that is buffering the excess of calcium that otherwise, if it's sustained, could lead to excitotoxicity. So we have, in this situation, both things: the calcium buffering calcium requirements, or energy supply and the huge complexity of these cells make the neurons to the need of regulate a mechanism of transport these mitochondria and distribute them all around the neuron where it is demanded. But once the mitochondria is distributed in the neuron as well needs a system to keep these mitochondria healthy and functional. And for doing that, there are two processes, fusion and fission, that allows the interchange of mitochondrial material and thus allowing the renew of the components of these mitochondria. But when a mitochondria accumulates too much damage, the neuron also needs another mechanism to recognize this mitochondria, identify this mitochondria, isolate from the rest of the mitochondrial net, and target it for degradation in a process called mitophagy. It is now we are currently understanding the molecular basis of these different processes, but there are still a lot of questions that remain to be answered regarding the regulation of these processes and also the intercommunication between them. In this context, we identified a family of proteins that are regulating transport and maybe some of these processes. But let's start for the origin. Initially, we performed a subtractive hybridization assay with the aim to characterize genes that were specifically or enriched in the upper cortical layers of the cortex during its evolution, meaning that they could have a role in the development of these structures. With this technique, we identified one protein, ALEX3, that with 6 armadillo domains in the C-terminal region and a putative mitochondrial targeting sequence in the N-terminal region. These features made this protein interesting for us to study. And we found that this protein, well, the name of this protein is ALEX because the previous identification in another study of 2 other members of this family that were shown to be lost or deleted in epithelial cancers that were originated from epithelial cancers. Our study demonstrated that this family was, in fact, for about 6 members of the family, not only 3. And what was the most interest for us was to observe that this family of proteins that is clustered in the chromosome X was only observed or was exclusive of the most evolved mammals in the Eutherian class. And we didn't found, or it's not possible, these proteins are not in other organisms. The origin of these proteins, or these genes, came from a retrotransposition from an ancestor gene, Armcx or SVH, from the chromosome 7 in humans to the chromosome X, and then successive tandem duplication gives rise to the whole family of proteins, genes, as it is known now. Interestingly, all the family of proteins is sharing, as ALEX3, these 6 armadillo repeats in the C-terminal domain and the putative mitochondrial localization signal in the N-terminus domain. The analysis of the expression of these genes showed that all these genes were enriched in nervous system in early stages of development and also in posterior stages of development in P21 on adults and localized in larger structures, although the expression was wide and general. Interestingly, the MIG-target versions of these proteins, when overexpressed in cell lines, all of them targeted the protein to the mitochondrial compartment, meaning that the putative mitochondrial localization signal found in the N-terminus is functional. We further investigated the role of these proteins by using the one that we observed or we obtained in the subtractive screening, and we observed that the protein, when overexpressed, again showed mitochondrial localization. This localization was in the periphery of mitochondria, finding that was supported by a recent published paper that showed that this mitochondria is in the outer mitochondrial membrane. The analysis of the endogenous expression showed that this mitochondria is localized in 3 different compartments, in the nucleus, in the mitochondria, and also in the cytoplasm, meaning that this protein could have different rules in these different compartments, or also meaning that it could be a communication between these compartments. During these experiments, we noticed that the overexpression of ALEX3 in cell lines and also in neurons was often associated with some sort of alteration of the mitochondrial net that eventually lead to the complete aggregation, and all the mitochondrial net was found collapsed in the perinuclear region, as you can see in this electron microscopy micrograph. This finding this phenotype was also observed when the levels of proteins that are related with mitochondrial transport are altered, either by overexpression or by silencing. And we hypothesized that maybe this protein could be part of this regulatory mechanism. To test this hypothesis, we conducted time-lapse video imaging with the overexpression of the GFP target version of ALEX3 and observed the chymographs from these videos. We could analyze and demonstrate that there is an important decrease in the number of mitochondria that is moving when ALEX3 is overexpressed. Interestingly, this decrease in the movement of mitochondria was affecting either the anterograde and the retrograde direction. And also, what we could find is that this alteration was also accompanied by alteration in other parameters of mitochondrial movement, like velocity and mitochondrial length of mitochondrial run. In a complementary set of experiments, we designed a specific shRNA against ALEX3, and we performed the same kind of experiments and analysis. And we observed that the downregulation of ALEX3 was also accompanied by a decrease in the number of mitochondria moving through the axon, and this effect was affecting, again, both the anterograde and the retrograde direction. What we observed, and I think is interesting, is that the other parameters, the velocity or the length of mitochondrial run, were not affected by the downregulation of ALEX3. And with all of this together, we hypothesized that maybe ALEX3 could be forming part of the molecular complexes that are regulating mitochondrial transport, but not the activity by themselves. And the effect that we are observing in the overexpression of ALEX3 could be due because of dominant negative effect of overexpression. To test the hypothesis of a possible interaction with this protein, with the molecular proteins or the molecular complex that is regulating transport, we perform co-immunoprecipitation assays with the different candidates, the different known proteins of the anterograde transport machinery. And we observed that indeed ALEX3 co-immunoprecipitated both with MIRO1 and with MIRO2. Also, we observed that when they were co-expressed, we both also co-immunoprecipitated with TRAC2. And finally, we couldn't find any interaction in the case of the kinesin motor, KIF5C. As you may know, it has been recently published a mechanism by which the neurons are able to detach mitochondria from the microtubule tract near the synapses, where the expression of the targeting of these mitochondria is needed, or the provision of energy is needed. And this mechanism is dependent on MIRO1 recognizing the calcium when there is a calcium influx after synaptic activity. We wondered if ALEX3 interaction with these components could also be regulated by the levels of calcium. And we performed the same kind of experiments, the co-immunoprecipitation assays, but this time in the presence or absence of calcium. And we observed that indeed the interaction of ALEX3 was blocked with MIRO1 and with MIRO2 when the levels of calcium were increased. This effect was also exactly the same when the overexpression of both proteins or the interaction was blocked when there were presence of calcium. Thus meaning that both interactions are sensitive to calcium. And finally, and as a control, we observed that the overexpression of the EF mutant hand of MIRO1 that is not able to sense calcium was interacting with ALEX3 either in the presence of calcium as compared in these two regions and this. So, meaning that this interaction is likely to be produced. But moreover, we wanted to see if this effect or this regulation of calcium, of the interaction between ALEX3 and MIRO, was also true in a more physiological range of concentrations of calcium. And to do that, we performed pull-down assays from mouse brains with against the construct, against the proteins MIRO1 or the mutant version of MIRO1. And we observed first that ALEX3 does interact with MIRO in the endogenous ALEX3 does interact with MIRO, and that in physiological situations, in a more physiological situation of calcium concentration, this interaction is blocked. Altogether, allow us to propose a model by which ALEX3 would be in the membrane of the mitochondria, interacting with MIRO and TRAC, and thus regulating this transport, and that this interaction and the function or regulation that is doing ALEX3 with this complex would be also regulated by the levels of calcium. Finally, we targeted or we mapped where the interaction was doing between ALEX3 and MIRO and the components of the machinery. And we generated a deletion construct of the C-terminal domain without the armadillo repeats, and we performed the same kind of experiments, showing that the interaction was dependent on the armadillo repeats. This is a very interesting data because, as I presented before, all the family of all the members of the family has the same kind of armadillo domains in the C-terminus domain, and that means that in potential all of them could be part of this transport or could regulate this transport. To test this hypothesis quickly, we did the same kind of experiments with the founder member of the family, Armcx or SVH, and we did the coin-Moore precipitation assays, and we found that again this protein was able to interact with MIRO1 and with TRAC2 and was not able to interact with KIF, thus meaning that the functional assays associated with these experiments, in where we also did time-lapse imaging of this protein, and we observed the reduction of the transport in both directions without affecting the velocity. So this is a very interesting finding, but we wanted to widen a bit the field, and we are now, and in the lab, they are now increasing the candidates to see more interaction of Armcx or also ALEX3. And I have been allowed to show this. Is a very preliminary experiment, but it seems that there is interaction between ALEX10 and also the mitofusins. And I know that they are also doing experiments with DRP, with other components of the known machineries. Importantly, it seems that with Parkin, there is some result that would relate this protein with mitophagy. So I think it's a very exciting field. And just to finish, just to show that this new cluster of proteins that are also in eutherians, meaning that a higher degree in brain complexity could also mean or be associated with a higher degree in complexity of the regulation at the molecular level, and places this family in the center, or with a potentially in the center of the regulation of transport and other processes that mitochondria needs to keep neuron activity healthy. So that's it. This is a lot of work, and it's done by a lot of labs, a lot of collaborations going on. I would like to thank Javier García Sancho, Antonio Andreu, Ramon Trullas, and Jordi García for the helpful collaboration they did. All the members that have been implicated in this project, all the members of the Professor Eduardo Soriano, and mainly Eduardo Soriano, because has been very supportive and implicated during the whole project, and Roman Serrat, because he's also a co-authorship of this project and has done an amazing work in it. And finally to CIBERNED to support the project. Thank you very much.
Thank you. We have time for one short question.
Do you know if Alex3 or any member of the family is up-regulated or down-regulated in Alzheimer's disease or in another neurodegenerative diseases?
No, we don't know. If it's, we tried to find the link between Alex3 and pathological situation. We did. We looked for mutations, possible mutations in Charcot-Marie-Tooth type 5 that is familial and is X chromosome linked without any success. And I don't know if in other pathological situations could be interesting to see any or look for any mutation in one of these members in any pathological situation.